Okay, so hello everyone. My name is Stephanie Burke, and I'm a postdoctoral fellow at the University of Toronto in Dr. Masi Moedi's group. I'm very pleased to welcome you to the very first Pain Research Forum Journal Club, and we're really excited to kick off this new series as well. So the idea here is pretty simple. I'm sure you've all been to a journal club before. Once a month, we'll get together and we dig into an interesting paper and hopefully have some good conversations about them. So this is open, of course, to researchers of all levels, and we encourage trainee participation and conversation because that's how we'll get the most out of this. So this journal club is part of a growing series that we have. So we're also doing webinars as well, which you might have seen on the slides when you first joined the webinar here. And basically, every month now we'll be doing the journal club, and each special interest group of IASP are going to take the lead on that session. So next month will be hosted by Courtney Bannerman at Harvard from Harvard, and she'll be leading the neuropathic pain SIG. Um, so yeah, feel free to put any comments, suggestions, or just questions or anything into the chat, or if you want to raise your hand, either. I think Adam there from the ECC of the PRF is on standby to keep an eye on those things as well. So yeah, you can do it throughout the presentation as well, or you can hold off until the end. Either works. So I'm representing the neuroimaging SIG today. I myself am relatively new to neuroimaging, so I'll do my best to represent this paper as best as I can. But again, if you have any comments, please feel free to drop them in the chat. So the paper I'm presenting today is cortical and subcortical mapping of the human allostatic interoceptive system using a 7 Tesla fMRI. So, acute stress is a vital evolutionary adaptation that prepares the body to respond quickly to immediate threats. It enables survival through the fight or flight response. And when we're exposed to acute stressor, the body activates its core stress response systems, most notably the neuroendocrine system and sympathetic nervous system. So these systems operate through key pathways such as the hypothalamic pituitary adrenal axis and the sympathetic adrenal medullary axis as well, coordinating the release of hormones like cortisol and adrenaline as well to kind of initiate this fight or flight response. So throughout life, we're exposed to this wide variety of physiological and psychological stressors, and they can range from, physical injury and infection to also the psychological stress as well. And these events can accumulate over time. So this tightly coordinated response system has traditionally been observed through the lens of homeostasis, which was first introduced in the 1920s. So homeostasis is basically that health is maintained by keeping these physiological parameters within narrow, stable ranges. But what happens with prolonged exposure to acute stress? So, to more accurately reflect this dynamic and integrative nature of physiological responses to stress, it was the concept of allostasis was introduced in the late 1980s. And allostasis is not a condition or a state of body, but rather it's the process by which the brain is efficiently coordinating and regulating the various systems in the body. So it's essentially adjusting the physiological set points in response to environmental or internal challenges. But chronic stressors, whether they're physical or psychological, they accumulate over time and they create wear and tear on the system or an allostatic load. And this is the cost of the physiological cost of the repeated activation then can overwhelm the body's capacity then and lead to systemic dysregulation and disease as well. So, in context of pain, which we're all interested in, so pain and stress have this bidirectional relationship and they interact on multiple levels, and there's a great deal of interplay between the two of them. So, individuals with chronic pain often have stress or stress related conditions as well, and they often exhibit elevated biomarkers of allostatic load. So the allostatic load model helps explain why psychosocial stressors, adverse childhood experiences, and socioeconomic disadvantage, which all increase the allostatic burden, are significant risk factors for developing chronic pain. And this conceptual framework emphasizes the chronic pain is not merely a sensory phenomenon, but reflects a broader dysregulation of interconnected physiological systems as well, struggling to maintain this homeostasis when it's under sustained demand. So the brain then is continuously anticipating the body's energetic needs, and it prepares to meet those needs as they before they arise. So that's what I've been talking about so far. So allostasis. But to support allostasis, the brain is continuously modeling the body's internal processes, and this is known as interoception. So, interoception is basically the sensing of the internal body states. So, this can be from temperature, muscle aches, thirst, and of course, pain. And structural deficits are involved in the interoceptive processing, or structural deficits in areas involved in interoceptive processing have been reported in those with chronic pain, including reduced gray matter volume in the anterior insula and cingulate cortices. And we know that the anterior cingulate cortex is involved in the effective motivation of pain, but it also serves as the primary hub for interoception. So individuals with chronic pain as well demonstrate altered interoceptive accuracy, awareness, and sensibility, including both heightened attention to certain bodily sensations and paradoxically then reduced accuracy in detecting physiological changes. So the kind of background for where this study came from actually was from the same research group back in 2017. They used a resting state functional MRI in almost 700 human participants and scanned using the 3 Tesla and identified this unified brain system that's integrating this allostasis and interoceptive system. And they also found that this is consisting of two well-known intrinsic networks, the default mode and the salience networks, and that these overlap in many of the cortical regions. So the default mode network is essentially interconnected brain regions that in the absence of external stimuli, it switches on or it defaults to internally focused thought processes. So this could be self-reflection, daydreaming, recall of personal experiences, and thinking of the future. And then in terms of the salience network, it's more about keeping an individual in homeostasis by selecting external stimuli that are deserving of our attention, essentially. And while this paper was very good and highlighted this network and the system, there were a couple of limitations that kind of fed into, I think, the development then of the current paper that I'm presenting today. So the limitations were mainly that the brainstem nuclei were not included in the analysis, and this is due to, I guess, inherent limitations with using 3 Tesla imaging. So you're limited in terms of signal dropout, the air tissue boundary, and the lack of regions of interest, or the ROIs, which I'll also refer to as seeds later on in the presentation. So really the aims for the paper even that I'm presenting today was to extend and replicate this allostatic and interoceptive network originally identified by them, and to use the 7 Tesla imaging. So it's a much more powerful neuroimaging technique with increased spatial resolution and improved signal to noise ratio. There are limitations to 7 Tesla as well, but not really touched on in this paper. So the methods for this study, they recruited 140 participants, but of that, 50 participants were excluded from the analysis, so that the final sample size was 90, and of those 90 participants, 40 of them were females. The reason for the exclusion criteria was actually quite broad. 19 withdrew before the MRI session even occurred, and then a lot of other kind of things cropped up. So due to discomfort in the scanner, incomplete scans, reconstruction issues, technical issues as well. So which led to such a big exclusion within the sample size. So within the imaging itself, they used the 7 Tesla imaging. They got a T1 structural scan, resting state. They performed 3 rounds of 10 minutes each, and they had diffusion weighted imaging as well. All of the pre-processing that they followed were using standard pipelines. And then for the connectivity analysis, they used basically a bootstrapping. So for the total of the 90 participants, the researchers randomly selected 80% to analyze, and then they repeated this process anywhere up to 1,000 times. So this increases test reproducibility, increases the confidence in the results, and then it also reduces the risk of false positives as well. So they generated positive connectivity maps as well with a significance threshold at p of less than 0.5. And they calculated the seed-to-seed correlations using Pearson's OR. And for the network identification, they constructed similarity matrices from Bootstrap positive connectivity maps. So you'll notice that, as I mentioned just there in the connectivity analysis, that everything was positive connectivity. And they applied optimized k-means clustering algorithms then to identify the default mode and the salience networks. So looking at the results, the first couple of results are based on their previous findings. So in figure 2, you can see that the authors demonstrated that the cortico-cortical connections previously identified at the 3 Tesla were replicated in the 7 Tesla. They also identified reciprocal intrinsic connectivity between several of the and the cingulate subregions, as you can see here. And they extended this prior anatomical findings from non-human track tracing studies. So the seed-to-seed connectivity or the seed-to-seed analysis confirmed basically 100% of known monosynaptic cortico-cortical connections that have been previously documented in animal studies as well. So you can see then in the left column, this shows the cortical seed locations, and on the right column then shows the group level t-value maps. So these were masked by voxels that showed the positive connectivity, and then with the seed in more than 950 of the Iterians from the bootstrap resampling. So when you look then at the figure up in the top right, so this is a heat map that is summarizing the strength of the functional connectivity between every pair of these cortical seeds. And what we see then is there's a robust positive connectivity across nearly all the pairs, indicating that these regions form a coherent network. Importantly then, the strongest kind of correlations occur between these insula and cingulate regions, which align with the known monosynaptic pathways linking the interoceptive cortex and visceromotor cortical areas as well. And so as well, this I suppose this structured pattern then supports a biologically meaningful organization as well. So then afterwards, they overlap the connectivity maps across all the cortical seeds, and the authors identified a set of connecting hubs that are kind of consistently involved across the system. So many of these hubs are then part of the brain's rich hub club, which you can see at the bottom of the right-hand side of the screen. And this suggests then that the interoceptive and allostatic processing are embedded within the brain's highly connected regions, which is what the rich club represents. So figure 3 then again is kind of replicating previous findings. So it shows that these cortical allostatic interoceptive system is organized into the large scale networks and basically how they converge on those shared hubs. So the authors, again, thresholded the positive connectivity maps from all of the cortical seeds, and they computed these conjunction maps that you can see over here. And then with the connectivity profiles, they were able to compare the resulting clusters to canonical cortical networks. So you can see then on the left-hand side of the screen, on the left panel, that network 1 resembles more of the default mode network, and network 2 represents more of the salience network. But what's quite interesting is in panel B, they computed the dice overlap between the network maps and the Yo7 network cortical parcellation using the network correspondence toolbox. So network 1, again, you can see is most associated then with the default mode network, and then network 2 is more associated with the salience network. And this basically provides a quantitative validation then that they identified allostatic interoceptive networks align with these canonical brain systems rather than, I guess, representing a novel pattern. I'm not sure if anybody here has used that toolbox. So the network correspondence toolbox allows researchers to examine and report the spatial correspondence between their novel neuroimaging findings and compare it then against the functional brain atlases available. So this toolbox was actually just published last year. I have the paper up on the top right hand of the screen if anyone's interested in taking a look at it. So it basically determines the magnitude and statistical significance of the correspondence among your user-defined maps and those existing atlases. From what I've seen, I think it's a good toolbox, which makes it easier for network neuroscience researchers to report their findings as well in a standardized manner, and hopefully then increasing reproducibility and facilitating comparisons between studies as well. So I look forward to exploring using that toolbox at some stage as well. So this is really kind of the whole aim really with the 7 Tesla is to extend those cortical findings by examining the subcortical and cortical intrinsic connectivity. So these analyses were enabled by the newly delineated subcortical seeds and the improved spatial resolution then and signal to noise of the 7 Tesla, which previously made these kind of analyses really difficult with the 3 Tesla. So using the cortical discovery maps and the seed-to-seed correlation analysis, the authors were able to replicate 90% or 96% of the known monosynaptic subcortico-cortical connections that were reported previously in the animal literature with the track tracing studies. So the main exceptions that they didn't reproduce were the PAG and insula and the parabrachial nucleus to the subgenual ACC as well. But this may reflect more of technical limitations as well, rather than not actually seeing these, because again, it's still relatively difficult to get these signals accurately within the brainstem. So each brainstem and then subcortical nucleus was used as a seed, and the resulting cortical maps then show reliable coupling with key interoceptive and visceromotor regions, including the ACC, insula, and as well as the medial frontal gyrus or the prefrontal cortex. So although the connectivity from some smaller brainstem nuclei appear weaker, as you can see in the heat map again here, in the consistent cortical clusters are still visible in the maps and remain robust across the bootstrapping analysis. So likely reflecting signal heterogeneity within the ROIs rather than the actual absence of this connectivity. So the seed-to-seed connectivity matrix as well demonstrates that most of the subcortical regions are functionally connected with multiple cortical nodes. And the conjunction analysis then as well reveals that the subcortical inputs converge on a specific set of cortical hubs, many of which overlap again with the rich club hubs. And suggesting an organization or hierarchical subcortical cortical architecture within the allostatic interoceptive system. So using cluster analysis then, they identified three functional subgroups. So lower brainstem nuclei were connecting primarily then to the posterior cingulate and sensory areas, and then the upper brainstem nuclei were connecting to the hypothalamus, to the mid-cingulate cortex, and then the larger subcortical structures as well had a widespread cortical connectivity. Um, I think for this paper, there was a lot of strengths to it. I'm sure if many of the readers would agree. The 7 Tesla significantly improved the resolution and provides this superior spatial precision and signal to noise ratio compared to the 3 Tesla studies. Again, as we highlighted there with the brainstem in particular, they performed the rigorous bootstrapping approach as well. And they were able to validate anatomical atlases as well of the brainstem. In terms of the scientific impact as well, it showed great translational validity with 96 to 100% confirmation of these known anatomical pathways from the track tracing studies. Also, comprehensive kind of whole brain mapping again, integrating the cortical, subcortical, and brainstem components of this system that they had previously identified. And as well as that, the rich club overlap as well, suggesting that the system and processes are embedded within the brain's main communication architecture as well. And of course, then it also has clinical relevance. In terms of, I think rather than limitations, there's more constraints to when you're doing this kind of work. So in terms of the methodological constraints, the resting state functional MRI reflects functional connectivity, but not direct pathways. The brainstem nuclei as well, they showed weaker signals, due to, I suppose, limitations as well at that level. And then the missing connections, but again, as we discussed, that this may as well be more technical limitations. But it definitely offers opportunities for future investigation, in particular to include interoceptive assessment. So, including direct measures of interoceptive accuracy, sensitivity. You can even include, questionnaire kind of data to this as well, or you can include tasks. And so, yeah, looking at the behavioral correlates as well, linking connectivity patterns to individual differences as well, if you can have the sample size for looking at these individual differences. So tying it back to pain as well, because it's been a lot of kind of heavy imaging stuff, information. The paper shows that the neural circuits supporting this kind of pain-relevant bodily regulation are deeply embedded in this brain-wide allostatic interoceptive system spanning, the brain subcortex and cortex. And that pain processing isn't necessarily just isolated to these pain regions, but it could be integrated as well within these networks that predict and regulate the body's energetic state. It's kind of a whirlwind, but I would.
Really beautiful presentation, Stephanie, and really beautiful work that you covered. So we can go ahead and open up discussion. If anybody has any thoughts while reading this paper or during Stephanie's presentation that you'd like to discuss, this would be a great time to do it.
I might stop sharing just in case people turn on their cameras. Okay.
I
Oh,
think
very
from my end, this is clearly much different than what I typically work on. So excuse me for any mistakes, but um, you mentioned that we were able to replicate, or they were able to replicate many of the animal tracing studies,
good.
which is super encouraging, I guess, and can possibly lead to future directions, right? Like functional testing in the animals. Now that we know that these networks actually exist in human.
Yeah, absolutely. Um, it's interesting. I think more and more work is being put into now using those kind of tracing studies from originally the animal models in neuroimaging. One of the researchers in Massey's group as well was able to translate neural circuitry as well from an animal model into humans as well. So it's nice to see this happening because I know it's quite difficult to achieve. So looks like we're kind of at that at that stage.
Yeah, good timing. Other thoughts from the audience? Lots of applause I saw go through. That's good.
Yeah, we might increase engagement as the time goes on. Everyone might be shy for the first journal club.
Yeah. Well, thank you so much for launching this first journal club. Everyone, please come back for next month's webinar. Stephanie is taking the lead at ISP ECC along with Tyler Nelson and running these for us. And like she said, we're going to go through the different SIGs at ISP. So hopefully we all get a taste of the kind of research we do and we can all kind of expand our horizons. Stephanie, do you have anything else you want to add?
No, that sounds good.
All right.
Thank you very much, everyone, for joining.
Have a great day, everyone.